this, like, we have lecture 8, 9, and 10 left in the class, and it's gonna be like really sort of, it's not gonna be like connecting to each other like the one we had before, but it's gonna be more applications of systematic and what systematic can do for you. And this one is such a thing that we kind of revisiting what we have talked about earlier, which is species concept. And today we're gonna talk about what I use the word delimitation. So the word delimit, so limit is limit, right? So it's no need to translate it. D mean down. So it's not like you put down like you leave the limit out, but delimit is to draw the limit. So we're gonna draw the limit on species, and this is not a calculus class. Or to be honest with you, I completely forgotten what limit is from calculus. I know it's in calculus, but that's all I remember. So I remember I have to find it, but I never know how to. So I only took one calculus class. So anywho, so like what we're gonna do today is kind of draw the boundary where the species are. We talk about how arbitrary the species are, right, back before the test, and we kind of know that it's kind of depend on what definition or what species concept you're using, and that is understandable. So we're gonna try to find a better way to do that, and a couple of different ways. I'm gonna point out like the ways where people have used to delimit, to like draw the line, like where you decide Thailand's end when the Lao start, like by the river, right? So, but the species are not that clear. They don't, as I said earlier, they don't really know their names, different things. So it's us, it's for most for our sake. And then the second half of this lecture, we talk about what we call, not I call, but what we call DNA barcoding, which is just like the name read. So you can read the barcode by DNA, you know exactly what species you're looking at. So sort of like once we have the limit of species down to nail, then we can start do something really cool like automate identification with DNA barcoding. So let's revisit this case a little bit. So we talk about this in my lecture about phylogenetic species concept and how this crazy set of lichens, Graphescripta, look kind of different, but not really. Are they like one species? Are they more? Are they like whatever? And I show that there are multiple specimens that are quite similar morphologically, but genetically distinct, right? We know from the phylogeny that it's 5 groups, but then looking back at these lichens and like you have no idea how to tell them apart, like you'll be like, of course I don't know how to tell them apart because I'm not a lichenologist, but you go ask lichenologists in Europe right now, and like if they can tell me each of these 5 groups of lichens are, I'd be impressed because I don't think they can. So that's what we call cryptic species. So that's the definition. So cryptic, just in the crypt, right, is like hiding something. So it's kind of hiding species, which some people feel like it's a misnomer because, well, I mean, they try like they're hiding because on the plain side they look really similar, but genetically speaking, they're completely different things. So deep down by blood, like well, like it doesn't have blood, but anyhow, you know what I mean. By the gene, their DNA, they know the difference for each other, but we are too stupid to see that. They were like, think of the monkey in the zoo, really. It's like you go to the zoo, right? And you're like, oh, all the monkeys are the cute. But that one look like the other one. And they probably feel the same way with us. Like you all look the same. You all look kind of funny, less hairy version of us. So the same thing, like we probably not have the eyes. They're probably different, but we don't have the eyes to see the difference. So we call them cryptic species. And so for some people feel like calling cryptic just really human centric because just because we can see it doesn't mean they're really hiding or anything. So that's one way to look at it. But anywho, so these are 5 genetic clades and they all over the place morphologically. And I had to look and look and look and look right before I found one characters that not separate 5 groups, separate only these 2 groups here. And you see this kind of graph in our case study with Matt Gilliams on that little flowers. And even this one tiny character can even separate species 3, 4, and 5. So what I did with that paper, if you're keen to read, you're welcome to, but I don't make any new taxonomy conclusion. Basically, I just keep them, you know what? They're all graph descriptor. Back to the basic, they're all like just one name. I don't make, I could be the splitter, and I think I told you already, I'm more like a lumper, and I hate it when people try to split things that not even visible. I mean, they probably like genetically they're like 5 different groups, but then if I'm gonna give you the manuals of telling these 5 species apart, you'll be like, what? Do I have to extract DNA and make a phylogeny? Forget about it. They're all just that species. So in the way, at the end, I go back to like, what would I do as a taxonomist, or what would I do as a normal biologist, try to identify this out, right? See, why don't you want to add your name to the species? And that's a really interesting ethics moment here, teachable moment, right? So the unspoken rules of taxonomy is that you not name anything after yourself. It's a bad test, really, really bad test. Just like, like you found a species, like, oh, I mean, like, I just named this lichen crayfish Crayshaki just for the heck of it, right? And people be like, oh, that guy is so full of himself. And it did, and it believe me, it did happen. It was embarrassing because you know why that happened? In Thailand, there's a paper from Thailand. The first author named the species after himself, and it's I forget what his name is, but let's say I do this like Graphish Crayshaki, and then I have Crayshak underneath it again. So it's like how how narcissistic you have to be to see your name twice on that species name, and for some people it's like not enough. So that's a bad test. So don't do that. Don't ever do it. If you get to name the species, you can do this. We call this technique scratch each other back. So like you name the species after your friend and your friend after you, and after some point it kind of come around. It's like, oh, like thank you very much. I didn't know that coming, but I named it first for you, so don't forget about that. It did happen. I seen that in action. Is kind of go around a circle. It's kind of comical, but I mean they did it for good reason, because they found so many new species. They run out of names to give. So what they do, so they auction off the rest of the name actually, and I think the highest they get is $600 for one name. It's lichens. It's not like Plutos or new mammals. So actually you can make some money out of it. So business idea, new startup, find new species, and like I'll name you new, I don't know, algae species, right? For $600, you can get your own scientific name, and then later on, the other one come in and synonymize your name to variety, and you're like, oops, sorry. So, well, I'm digressing. So, so that's what happened. So you'll be like, okay, fine. This stupid lichen have 5 species and we can't tell them apart. It's cryptic species. Why does it matter? Like, why do we bother with this cryptic species? Like, nobody gonna care about lichens, right? I do. So, but this is something that you might want to care if you don't care right now. So this is your friend's Cryptococcus neoformans variety gaddii. Is used to be clinically, if you're a medical technologist, this is just one species. Under microscope, they look very distinct, right? Just circular blobs. Like I would not be able to tell them from, and you can tell from the name, right? Cryptococcus. Taking microbiology, coccus is a circular bacteria, and crypto mean cryptic, like hiding circle thing. So thank you very much. Your name is not informative at all. So, but this thing is really bad because it's a fungi that cause infection in your body, and they're quite prevalent. They're not lethal per se, but they cause you, they make you sick. And we do the phylogeny. It turned out all the things that we call neoforman variety gaddii actually split out further into 1, 2, 3, 4, 5, 6, 7. There are 7 species hiding in that neoforman gaddii looking thing. So morphologically things that look like that actually have 7 different genetic groups. And you'd be like, okay, sure, they have groups, and then what's the matter, right? Why do we even bother? Because this is all these different scriptic species respond differently to the drugs we give them. So this is what they call the phenotypic screening. They give so all these different antifungal treatment, and plus and minus is the response they have to each of the drugs. And you can see that all these 1, 2, 3, 4, 5, 6, 7 used to be all one species, and they respond to the drug differently. So this one, like if you have deuterocati, they will not respond to the DL lactate you give them. So like, oh yeah, like that's nasty, right? So and they also give in to different dose of the drug. So the moral of this story is that, like, by not being able to tell the species apart, whatever the species are, you're missing out on their biology. Some of the species group might not matter too much. They probably like, just like lichens, it probably won't affect your life. But things like this, you want to know exactly what being treated, right, and how they respond differently. And it could have updated with the most recent data that we just heard about, right? All these COVID viruses have different variants, and different variants respond differently to the vaccine. And they're not like, it's not that they're getting worse and worse. It's more like, well, they're not called Indian and British anymore, called Alpha and Delta. So see, the naming comes to haunt us again. So the Alpha did not respond well to the existing vaccine, but the Delta, the Indian one, it turns out from what I read this morning, like most recently, they seem to respond to the existing vaccine pretty well, meaning the current vaccine can kind of keep up with it. But yeah, they use Greek, not Latin, so Alpha, Omega. So but the Indian the Delta strain like spread much quicker, so it's kind of like, it's a balancing act that they have enough immunity, but at the same time it like spreads so quickly. So like, which one gonna go first? And our prime minister is so confident they're gonna get the country back in shape in 120 days. So watch out for the next movie, 128 days, 120 days later. So good God, right? So that's why we should care about cryptic species. So why are species cryptic? So there are a couple of different reasons, and I kind of pull out some of the reasons that should be pretty obvious to us. So yeah, I'm not going to respond to that. So, the recent divergence, basically, if you think of is evolutionary trees, right? And phylogeny start diverging like very recently. So it's not enough time for them to be different. They probably start diverging already, but the difference is still very small, and us blunt to humans cannot see for what it is. You need to develop a finer eyes for those species. But we did not, well, they probably just recently split, maybe yesterday, and they would not see it coming. So that's first reason. And second reason is the environment. It could be environmentally constrained, meaning they under some selective pressure. Think about natural selection. Like, for example, if you want to be in the water, right? You kind of need to have a gills, or you have to be unicellular, have to have some way of surviving in the water. And the fish cannot be that different because they can't do anything else without gills, right? So you kind of need gills. So maybe that's why, like, it's an environment that kind of forced them to be that. And that character is not that different. So they sort of like, I want to be different from this guy, but I just can't, we all need to breathe. So I like, as a fish, I would love to have some new breathing apparatus, but it's apparently not invented yet. So I kind of stick with the gills. So as much as I hate to use the same gill as that guy, who are now different species from me, I can. So that's one way. Or the third way, actually this happened more often than the first and second way, is that you could have a species that diverging already and they become two different. So let's say they have horns and this one like have hairs, but then for some reason they converge back together, and that's kind of related to the second reason, right? They sort of and they come back to the same environment. The horn gets smaller, and this one the hair become the horn. So like, oops, you look kind of the same now. So now they are currently cryptic, but back in the evolutionary history, they're not. So it could be from convergent evolution. And that's a funny thing because evolution happens all the time. Evolution doesn't wait for us to get over with COVID for us to study them. You know, evolution moves on. They keep moving forever. So it just, I mean, you don't know, like, are you gonna catch them here? Are you gonna see them here? Are you gonna see them back here? You don't know. So for things that evolve faster than us or having shorter lifespans like bacteria, we can observe that in a lifetime. But imagine you study, I don't know, tortoise morphological changes, right? You kind of have to like wait until they grow old and have babies, and that could be the same time you have babies or even later. So it could take forever. So not possible with every single one of them, and that's why cryptic species is actually more um, common phenomena than most people think. We tend to take things by for face value, just like the case of giraffe. I think a lot of you mentioned that giraffe is a surprise to you because you think that giraffe have one species, but then like you look at the coat pattern, it's tiny differences, and we would not have known that had we not looked at the phylogeny. So those kind of cryptic things happen, and it is quite difficult to see. So how do we fix the problems? Um, we have two different way of delimitation. So we can do the good old morphology, and this is gonna be pretty equivalent to phonetics. Basically, like instead of just like, well, giraffe is long neck, like, okay, you know what? The small to giraffe. Giraffe is just not just a long neck. Giraffe is more than that. Like, don't read giraffe by its cover or by its neck, whatever that means. So you kind of have to give giraffe a fair try, right? So, okay, let's look at everything you have. Just measure everything. Measure the length of your ears, the first hair, and like measure everything and see if we can separate you. Or if you're too crazy, if you're too lazy about measuring morphology, you can use genetic information. So you can use genetic limitation, and there's two ways of doing that. We're going to talk about both of those. So in a way, we use all the information that we can to try to figure out the different species with in mind that there's a actual grouping exists that we don't see immediately, and we try to be more detailed. So that's the first example you already seen from the previous case study, right? So before that, these two varieties were in the same species, right? Hypogonella, Arizona, is Palmeri, and they separate into two varieties because people think that size doesn't matter when it does in this case. Size does matter here. So, and it took Matt and Tim for a long time to measure every single things. And if you recognize this, like they actually ever measure the appendage length, which is this. They measure this little hook on the fruit. It's just like, imagine if you were to do this for your thesis. It's like you probably hate yourself or hate the person who made you do it. So rightly so. And it's really minute difference to separate this apart. And then finally, until somebody come around and use really detailed morphological data to see this. So this is a case of like go back and rethinking about what is different between these groups. And then you can do is multivariately. So sometimes when you look at the petals and the sepal separately, they don't look different. You try that method already, and it doesn't pan out the way you want it. But when you look at things together, you combine all the variables together, you can see the grouping more clearly because you combine different characters together. and that's something that for a lot of us, it's not humanly possible to see that difference because the difference is so minute, or it's just a combination of difference. It's different of this by that, different with this by that, and you have to like put all of them together to see the big picture. So this is when phonetics come in useful, even though it feel kind of outdated, feel really statistical and non-biology, but at the same time, that's what like allow us to see the cryptic nature of the species because it's hiding somewhere we don't know about, if that makes any sense. So I mean, most of this would be sort of review for us, but kind of remind you again that this technique are useful also for delimitation. And let me tell you right now, there's a few people on this world, not a few, but like not that many people in the world who are willing to go this route because what is tedious as hell. So it did happen. I made my student do this and they probably secretly, quietly, ragingly hate me for that. And they didn't tell me, or they did tell me, I choose to ignore. So um, yeah, it's like to say PCA is already old, but you can do it with any other methods. And this is something that you are guys are either golden age of computational tools because there are now like 5 million tools to do the same problems. And it, like just the same problem when you hit the captcha where it's a traffic light, the same way like the computer can help you figure out the cryptic species. And there's a lot of paper now on machine learning of species identification. So you can take a pictures and they tell you this dog for sure and can tell you what breed of dogs. I tried those apps. They're not very good at this point. So they still can't tell the different cats and dogs apart, but they try. So but yeah, there's like so many techniques right now and they're just taking different ways and machine learning is fun. I'm learning it. So Tube have to be patient with me. I'm like, you know, I'm catching up with you. So all right, so that's the difficult way. And this day with the image analysis, you can get all this morphology data really quickly. And there's even in an app that like you take a picture of leaf and they measure everything for you, like automatically. Crazy. So I'm like, God, I feel like an old man. Not that I'm not old, but like I'm like feeling old right now because like things that you have to do by hand now is like automated. But then let's talk about things that make our life slightly easier. So the distant method is to basically is short for genetic distance. So instead of like labeling yourself with all those morphology data, they actually just look at the percent genetic distance or genetic similarity depending on who you're talking to. But basically, bacteria people are quite comfortable with this. They're like, you know what, guys, why are you fighting about? The genus are easy. If they're 96.4 similar, they're same genus. If they're like between 95 and 92, they're the same family. So yeah, easy. It's just like sequence the DNA, sequence the shit out of DNA and just compare them, plug in some numbers and like, here we go, it's the same species. And you ask them, because you just learned about nomenclature, like, so what's the species name? Like, I don't know. It's the same. The same as what? Same as the reference. What is it? What's the name? So it's the name of that reference. So the bacteria people have really like they kind of like forget about nomenclature. It's too much work. We have more important things to do right now, which is like curing disease and all this crazy viruses around the world. So they're like, okay, let's use something real quick. And then this way actually make it more precise for them because you see from the case of COVID, like one tiny little mutations have different symptoms, have different spreadability and all that. So I think for microbial people, this like makes sense to them. Like just use a similarity, you just compare the sequence and you'll be done. But then for us bigger organism people, that sound a bit too sheepish. Or I know like it's easy way out. Like you guys, it's just because they're gonna say doesn't mean they're related. It's like, okay, you guys thinking too much. But okay, so fine. Let's do it in more complicated way. So since we learned the tree, so the tree is basically come from phylogeny. right? And this is where I bring back the cladistic to you guys. So remember in cladistic, right? So this is under cladistic. In cladistic, if you are the same species and ask this in the test, why would tocogenetic relationship be different from phylogenetics, right? And this is because of the gene flow, right? So because of the gene flow in the same species, the individual of the same species still are somewhat similar to each other until this happened, until the reproductive isolation happened, and then the relationships start to diverge, and then they only have gene flow within the same groups, and those two groups become two different species, and that's phylogenetic relationships, right? That should be not strange to you anymore, even though this term probably still haunt you for some reason. But then if you look at the branching pattern, you will see something different, right? Because if you compare, oops, sorry. Uh-oh, why did you do that? Don't do that. Don't do that to me. All right. Maybe it's like vaccine thing starts kicking up. I start like making no sense. So if I start to like slow my speech, like yell at me on the chat box, and so I know that I'm like affected by the vaccine. I'm gonna blame all my mistakes on the vaccine right now. So, it's just so that I don't have to blame myself. So anywho, so if you were to compare the sequence from here and here, right? Let's say you compare this individual to this individual. The pattern would be so different than you compare this one to this one, right? Because this one would be more similar, and like the difference between them will be um, um, between the same species. So the branching pattern between them will be different. So GMYC, this method with really confusing names, generalized mixed U coalescence, or GMYC, is basically trying to find this line. So where is a different, like where is the tocogenetic relationship stop and the phylogenetic relationship starts? So that's why I asked you earlier in the test, like, why are they different? Like, why should be they different? Like, can we use the molecular data to detect the tocogenetic and phylogenetic relationships? And Raymond's talking about Bill and Melinda Gates Foundation. I don't know what it's supposed to be. Oh, it's it's from CPP. It's a Sinovac. So it's a vaccine joke that I didn't get. So nope, I did not get anything from Bill and Melinda except the news that they broke up finally. So I don't know since when Melinda and Bill turned into the interest of Entertainment Weeklies. So, but anyhow, everyone's breakup become Entertainment Weekly business. So, anywho, so let's look at this. So, for GMYC, you kind of like you have the trees, right? And there's like more than one groupings in there, and you sort of like it's pretty simple. You count here, so like there's one branch here, so there's one, and you slice here, there's like, oh, there's two branches, and there's two, and you keep going that way from the root to the tips. So imagine you have a tree, right, and you like cut the tree up in slice, right? So you just like if you the CT scan, it's like kind of slide your body up into smaller slices. And then me people, I'm sorry to remind you of your nightmare last weekend. So kind of slide out and count number of branches, and you do this like slowly, and at some point you stop here and feel like, all by a sudden there's so many branches. Oh my god, like there's so many branches, what's going on here? So you now realizing that you are entering the tocogenetic zone, right? This is within species, and before that is between species. So this is like, oh, okay, I cut here. So whatever, so this is just like cutting the trees, whatever left from this cutting, the grouping you see here is all different species. This is a lot of species actually. So that's the idea. So if you have difficult time figuring that out, I recommend that I should have done this. I just threw out my last broccoli. You go to supermarket today or have somebody go it because you're afraid of COVID. So just buy a head of broccoli and pretend that you know how to cook, right? Or watch a cooking show. And you want to cut the broccoli into smaller branch, but you don't want to cut them completely to the tip, right? That's just one tiny like dots. You don't want that. You just like want still nice bush but smaller. So you like find where to cut nicely so it's going to your stir fry or your salad in the appropriate shape. That's basically that. So same concept there. So I'm glad somebody know how do you decide where to cut. That's a good problem because some people like to eat broccoli big, some people like it small. So I mean, broccoli wise, I can kind of leave it up to you where I want to cut it. Oh, this tree. Oh, not broccoli. Anyhow, so what this tree does when they decided all by a sudden the number of branches start to like increase really rapidly. Or if you're a calculus based person, this is the inflection point when you like your curve is like it's like roller coaster, like the point is slow you down like that when you realize that that's the changing point. And if you look at the scale here, it's not the normal scale, this is log scale. So you go from 50 branches to 100 branch in like a small distance. So you know that something is happening here. So it's like a change from between species to the different species. Yep. And the algorithm basically does that, just like check the number of branches and then look for the uptick, basically. Yep. And this is like sort of very simple, like conceptually, but not computationally. I mean, you could, there's a code for it if you want to run, but there's some people who already wrote the program for us to do. I'm gonna try that today as well. So that's the first method in using tree to delimit where the species are. And the second method is, second type of method is called species tree coalescence. And this is gonna be a bit more convoluted because they kind of do this. So the word coalesce mean come together. So if the species split properly, and you define properly really broadly, depends what you mean by properly. So let's say this is like the one gene, right? So the gene, there's like the species B splitting from species C. So if you draw the phylogeny, it would look like this. A, B, and C. So we know for sure, let's say we know for sure that B and C are related and they split from each other. And this one particular gene do exactly that. The alleles match up with the pattern of species. That is good, right? That's fine. This one is called deep coalescence. Like this is the genes and species tree agree. No problem. Right? But they also know, because this is gonna get a bit more philosophical and it's gonna feel a bit more interstellar moment. It's like quantum physics moments. Like your body is not you. Your body is just like the organization of genes and organs and cells working together. At one point, when they stop working together, you die, right? So the same thing with the genes, right? In the same species, you would think that all the genes follow the species boundary, right? You would think so. You would hope so. You would hope that they all like follow the rules. Like we're now different species, we must split. And not all genes do that. Some genes are, they have their own history. So this particular gene, A and B split first, and then C come from completely different things. So if you were to draw the tree from this particular gene from just this one gene, your gene tree would look kind of like this. It would look A and B together and C, and that's just the tree from that gene, just that gene. So in this case, gene doesn't follow the tree. So in this case, has a name. It's called incomplete lineage sorting. So it's incomplete. So it's not sorting out the way it's supposed to. Like it doesn't sort out in the same slot as a species and have different branching pattern. And your body is just like 20,000 genes, and like when you decide, like it's not gonna be everyone gonna behave the same way. So, oops, what if you pick the wrong one? What if you pick the bad one to look at to this relationship and like, well, touche. Like really, that's your response, touche? Yes. A lot of times we've been into too many touches and we're like, oops, we picked the wrong one and we picked the new data, we get different answers. So this is when the specialty correlates method come into play. So they look at as many possible genes or regions as you can get the data on. So for example, here there's 3 species we know for sure. There's 3 species of this L thing, right? And then you look at 5 different genes like CO1, ND1. This first 2 is mitochondria. And the third one, ITS2, the BG, and CAD, this is a nucleus one. And you kind of look at the branching pattern of all these markers. And if you do sort of like a big picture thing, you can see that it's sort of split like that for 3 of them. But then these 2 is only do 2 split. So not all genes have the same history, and you kind of average them out. So you sort in the really small, lazy way of saying this, you, and I use this word with like a lot of caution, is you averaging out the relationship, where should they be, and you get this what they call species tree. This is where the species should be related based on different markers. And it's sort of like scheduling people on the same calendar. You guys all have different, busy and free time, and you try to put everyone on the same room at the same time. It's like, okay, guys, tell us what works for you. Like, no, not Friday, not Wednesday, not Saturday, not ever. It's like just, so this species tree try to solve all this problem because deep down they know that everyone evolved in the same organisms. You guys should have some agreement somewhere. So this is computationally quite intensive, and they kind of try to find like the middle ground for all different genetic history. So sort of try to find agreement. It's not democratic. You might feel like, oh, so the first 3 genes had agreed that there's like 3 different the first 3, like these first 3 seem to have this answer, but then also they win, right? So they do this. No, it's not that. It's not even that. It's sort of like, I think I used a bad metaphor before. It must be in the Sinovac. So the proper, I think I found a better metaphor. So proper metaphor would be like, you are a cop, right? And you interrogate suspect, like 5 suspect, and have so tell me what happened last night. And some of them are lying, but all of them are at the same time, at the same place. So there's a truth somewhere out there, and you try to find that truth. You know that all these 5 people being in that room at that time, they see something. Somebody might be lying, but if you patch that story together, you get a good picture. So that's why when two ask me, use mean or median, they use neither. They use consensus or agreement among those different disparate history, hoping that they can get the true sense. Yeah, not even a union is strange. It's sort of like, when do they coalesce? Like when you light up the story, or like when does it work? So like all these 5 suspects talk about different things, and you're like, there's one common thread. Like they all talk about having party at 10 p.m. last night. So like, that must be true because they all say the same thing. So you can think of it as an intersection of the truth, and you try to find a common history, right? And I know this sound a lot of mumbo jumbles, and then the algorithmically, which is a really mouthful word to say, algorithm wise, there is a way to do that, and it's quite intensive because mostly you use Bayesian. So so that's for the math for smarty people. They can do that, and this, I mean, I run this couple of times, and then it's like take super long to do, even in a small tree. So, and this requires that you have multiple data set to work with. You have multiple genes and markers to work with. So that's kind of prohibited for some people, prohibitive for some people. And today we're not going to attempt this, but we're going to attempt the GMYC, the previous method. All right, so that's kind of like super quick and dirty version of species delimitation. I attached a paper, which I love the name, on Canvas. It's called How to Fail at the Species Delimitation. It basically a review of all the species delimitation methods in greater details, but I'm not gonna count on you to read that paper because it might be over most people's head, including mine. So, and I think the fun part is that they point out at the flaws of all the techniques. It's like all the methods sucks, by the way, but we have to use one. So pick the worst, pick the least sucky one, I guess. So, and the GMYC GMYC are one of the worst methods you can use, but it stay around for so long because of its simplicity and its ease of using. So, people like to be correct, but at the same time, they like to get their work done. So, that balance, like, let there be a problem for future generations, that we don't want them to out of work. It's just, we create more problem for them to solve in the future. So there's a lot more to this, and this discipline is really active. And as I said, there's like more computational tool this day. You can like plug in different computer programs to do this. And to tell you the truth, it's really that, again, I'll say this over and over, is that the organisms, the plant, the animals, the bacteria, the fungi, they don't know what they're being called. And they're not going to respect your time and energy trying to figure that out. They're like, the way it is, I'm going to be really great, and you can never figure me out, haha. And that's the way it is. And for us, the species boundary are just for convenience. We want to treat the disease. We want to tear them apart. It's for us, really. Like, they don't care for 1 second what they call or if they save species. So this method try to capture the best possible units that we can use for systematic, because with our system, like biology, just a big mess, like this is why we have this class. We try to make some sense into the grouping, and we try the best we can, even though it's not going to be completely 100% correct. Welcome to biology. You'll never be correct again in your life. So not like maths people or physics people, they feel like they get the correct answer all the time. Like, nah, not with biology. Like, these are exceptions and stuff like that. So yeah, that's why we're jealous of them so much. So yeah. So that's a science, right? That's the beauty of science. You never know what's for sure. So even math, see, too says even math is undecidable. So there's so many unsolved things in math. So that's why we do science. So let's put that aside for a moment. I'm gonna